Hello, everyone. Welcome to our monthly Science of Alt Protein seminar. Today, we're going to talk about enhancing alternative protein through microbial diversity. My name is Adam Lehman, and I am a principal scientist at the Good Food Institute focused on fermentation. For those of you who are here for the first time, thank you and welcome. The Good Food Institute is a nonprofit think tank 100% driven by philanthropy, and our goal is to build a world where alternative proteins are no longer alternative. If you're looking to take a deeper dive into the science of plant-based, fermentation, or cultivated meat technologies, check out our open access resources at gfi.org/science. And with that, I'd like to introduce today's speaker. Dr. Shahin Ali is a senior scientist at ATCC, leading the bacteriology and mycology content team. He has over 15 years of experience in the field of fungal biology and microbiology. Before joining ATCC, Dr. Ali worked for the USDA ARS at Beltsville Agricultural Research Center, Maryland. He obtained his PhD from University College Dublin, Ireland. And today, we're going to talk about microbes. And while there may be a decillion different species of microbes on Earth, there is much work to be done to understand the appropriate species for alternative protein production. Today, Dr. Ali will speak to us about the ATCC's effort to leverage their resources to introduce new strains and species into the alternative protein ecosystem. Shahin, thank you for being with us today, and I'll turn the floor over to you.
Thanks, Ed, and thanks for the nice introduction and giving me this chance to talk, give this webinar. So I joined ATCC in 2022. It's almost 3 years. So I'm in the content and accessioning department. My main job used to bring new strains to the ATCC. But then when I looked into our existing collection, then I thought that how we can use it into this. And then I came across GFI, and we had a very nice relationship with Adams, Lucas, and GFI, and ATCC and GFI is working together. So today I'll give you some idea about alternative protein and how microbial diversity, how the microbes that we have in ATCC, how it can enhance this alternative protein industry. So basically to start with, so just to give you a recap about what is alternative protein. So as I guess all of you know that all this alternative protein include plant-based, fermentation-based protein, or cell culture meat-based protein, or even insect protein. So people are using this kind of protein for centuries as their food, but the modern alternative protein, what we are talking here is mainly different than those things, is that we are trying to replicate this taste and texture of the meat, okay? So it all begins in around 1960 with all this soya and wheat-based meat substitute, and then the Curon came in around 1980s, then Beyond Meat in 2012. So these are actually soya chunks. And I remember I grew up eating this. Like these are very popular. I'm from India originally. So these are very popular in India and some Southeast Asian countries. And I guess this is still very popular among the vegetarian population. But to be honest, these things taste really bad. They taste like rubber. I really don't like it. But so I was thinking that we can do a lot, some good justice to these kind of things, and we can make the taste and texture better for this. And then I guess all of you know that then the COVID came. When the COVID pandemic came, it significantly enhanced the popularity of alternative meat due to various like supply issue to the traditional meat and all this. So this also gives us another like an important point that when the traditional thing get disrupted, how these alternative things get the popularity. So today I'm not going to talk about the health and environmental benefit of alternative protein, because I guess all of you know about that. But basically, I'm going to focus on another issue, which is our our planetary health, the health of our planet. So basically, I guess all of you know this image. Like these are the 9 planetary boundaries that we need to protect if we want to protect this Earth for our future generation. And already we have crossed 7 boundaries out of those 9. So, and if you see in last 70 years, we have changed the face of this earth. Okay, our human population increased by almost 3 times. The forest coverage gone down by almost like 1/3. Then almost 75% of the wild population has gone. And then the land use for agriculture and like for special, like say meat production has increased by 36%. Okay. So definitely we need to slow down. We need to rethink how we're gonna treat this planet, because if we keep on going like that, there'll be a no planet for maybe our grandchildren, maybe in 2, 3 generations. So we should do various things like reduce greenhouse gas emission, like plant trees, but we should also change the way we eat. Okay, so we need to change our food, and because you see, normally food consumption contribute around 10 to 30% of total greenhouse gas emission. Now the question is, can alternative protein be part of this solution? Definitely. Like I guess you have seen a lot of publications, and people talk about this can be a good solution, and because alternative protein always have less carbon footprint and all those kind of things. But if you see practically globally, only around 5% of the total protein market is alternative protein. Okay, account from alternative, and US this is even less, only 1%. So the question is are people willing to change? Okay. Uh, and if you go with this sustainability message, it can appeal only a very specific niche of people. To be honest, most of the people don't care, and you cannot blame people for that, because it's humans are like that. Okay, we are selfish. That's because we need that's our evolution, how we evolve. Okay, you to survive, you need to be selfish. So we need to make this alternative protein acceptable to the mass, and so that now, for the sake of the planet, and to make this alternative protein acceptable to the mass, we need to improve the taste and texture. We need to increase the affordability and accessibility of this alternative protein so that price goes down. Then we need to focus on the health benefit, and we need to foster the cultural and culinary aspects. Like we need to fit them according to specific country, specific culture, specific culinary practice, these things. On the other hand, like there's my thinking that this is a choice versus necessity. Now, at the present, alternative protein is completely the choice. Okay, if you don't want to eat this alternative protein, it's fine. You have a lot of other choice. But I guess within few decades, the way things are going on, all this environmental change, extreme weather, and all those things, the cost of production for traditional meat will go up. Traditional agriculture will go up. It will become more like unpredictable, and the acceptance will go down. And on the other hand, the way we are developing all this research and development scale up, the prices for alternative protein will go down. It will become more and more acceptable. Okay, so I guess the time will come this when this alternative protein will be no longer alternative. It will become like a mainstream. But when that will happen, we don't know. But we need to accelerate this process. Basically, we need to develop alternative protein. We need to accelerate this process. And we need to prepare for this situation when the traditional protein will become less and less available and more expensive. And like the COVID-19 was a classic example. So today I'm going to mainly focus on how we can improve the taste and texture, and how we can make things affordable using different microbes. Like there are many ways people are trying. Okay, so the whole industry researchers, they're trying to do this one. There's many ways, but I think one good way is to use microbes. Okay, to improve these things. And if you see in terms of taste, so basically, if you try to improve taste, you need to add flavor, you need to add some custom protein or fat, and microbes can do that. We can use precision fermentation, we can use traditional fermentation. There are many microbes which produce fats. We can add that. And if you try to improve the texture, basically we need to make this alternative protein like more fibrous, flaky. We need to create scaffold, add blending, and add binding agent. And microbes can do that. Like you can add fungal mycelia. Microbes can produce a protein which helps in cross-linking different compound. Microbes can produce different gel-like formations. Then cost reduction, like one cost reduction strategy is to have a novel base protein. So this is like, so say currently most of these alternative protein are based on pea protein. Okay, which is again like agriculture based, it's vulnerable to all this climate change, weather pattern and all those. So if we can get a microbes like which can produce like an high quantity of single cell protein, which is cheap. So based on that, you can use that protein as a base, and you can keep on adding other ingredient to improve your flavor and texture and whatever. Okay, and then we need to reduce the cost. The whole process should be resilient and rapid and resilient. And microbes can easily, we can use microbes easily to achieve all those. Okay. Uh, like microbes can use low-cost feedstock like agricultural waste and all those, and microbial fermentation, it's easily scalable, or you can scale up, scale down depending on your need, and it is not much dependent on the supply chain and all this. And then fortification, because we need to improve the nutritional value of this product. We need to improve digestibility. We need to reduce the allergen and all those. And microbes can achieve that. Microbes can produce like vitamins, mineral. It can enhance the mineral availability. It can enhance bioavailability by degrading complex compound into easily digestible compound. And you can use microbes to tailor the nutrition. So like basically you can produce food like say for a diabetic people with high cholesterol, or with people which are allergic to particular compound, because microbes can degrade things and you can tailor it. So we have the recipe here. So basically I'm giving the recipe. So now the main thing is that now we need the ingredient, and the main ingredient is microbes. Okay, so the next question is which microbes to use and where we can get those microbes. So ATCC, in full form American Type Culture Collection, we can help here, because I guess most of you know ATCC, or maybe many of you don't know, because most of the time when I go to the conference and I introduce to the people that I'm from ATCC, they said, oh, you guys sell bugs. So yes, definitely we sell bugs, but we offer much more than just bugs. So I'll give you a recap, like a nutshell, what is ATCC, what we are doing, and how we can help in this aspect. So ATCC was funded in 1925. So next year we'll celebrate our 100-year celebration. So for almost last 100 years, we are leading suppliers of all these authenticated microbial strain, viruses, cell lines, and all this. We are also, other than that, we are also a service provider. We provide like various custom-based service, drug discovery, other things like CGMP service, and the biorepository service also. This is more relevant to this group, because if you have something that you don't want to distribute into the public, you can use our biorepository service, where we'll basically, like it's a safekeeping, we'll keep it as like a long-term storage process. Then we are also a patent repository. Almost more than 90% of the US biopatent related product are stored in ATCC. And all our product are all this ISO 9001 or all this ISO different certificates. So basically this gives you the amount of quality testing goes into ATCC product. So what makes ATCC collection unique is that it's our non-profit scientific mission. So we have a mission to preserve and authenticate all this strain, all this microbial strain for long-term benefit. Okay, we have strains almost for 100 years old strains that we have in our collection, and we are storing that. And we basically we don't care about whether these strains are in demand or not. Now, if you see from our collection, around like 10% of our collection only brings revenue for us. Okay, but rest 90% never sells. But we're still keeping those things, because that's our mission, because we never know that sometime those things may become important, or something may happen. Like say, for example, if there's a new industrial application, you might think that we already have those microbes with those metabolic capabilities. Or if there's a new disease outbreak, we may already have this relative isolate or relative species in the collection on which people can start doing research and like this. So one classic example I'll give you, this camembert and brie cheese fungi. I think many of you seen this news item coming like around early this year in February, March, that this French camembert and brie cheese are in decline because they're losing the diversity of microbes used for production of this cheese. So then we looked into our collection, and we have strains as old as from 1904. And now we are we are trying to collaborate with this French institute to test our strain to see whether we can use this strain to rescue these Frenchies. So this is just one example. I can give you a lot of other examples like other diseases like Zika or monkeypox. So this is the value of like keeping this type of collection level. So now let's go how we go through, because I already mentioned that our strain go through very stringent quality testing. So when we receive something, we do the genotyping, we do the authentication using genotyping, phenotyping, and functional analysis. And then we the way we do the QC, and then we produce safety stock, seed stock, distribution stock, the way we manage things, store things, that we minimize the passaging so that we minimize the phenotypic and genotypic drift of these strains. And currently we are relying more and more on the whole genome sequencing, because as you know, this is the era of whole genome sequencing, and this is very important in terms of food safety, regulatory compliance in this field. Because, like, say, if you file for a regulatory clearance with for something, you don't want to go back after a few months or a year saying that, oh, the species name has changed or something has happened. And especially when it comes to fungus, the traditional classification is very tricky, because fungus are like notorious. They have all this morphological similarity, cryptic species, species complex. They have all this hybridization, gene transfer of different ploidy level. So even like a multi-locus sequencing like IDS is not sufficient for species identification. So these days we are more and more relying on the whole genome sequencing to identify the taxonomy up to the taxonomy level. And we are relying on mostly like ANI based labeling, like average nucleotide identification, genome based ANI, to identify these species and classification. Like, for example, this famous fungus, Fusarium venetium, the Curon fungus, it has already gone through one name change. And Fusarium, as you know, is a species complex. It's it's very tricky to identify the exact species. So that's why from last few years, like I think in 2019, we started a ATCC genome portal. So this is very similar like NCBI genome portal, where you get all the genomes. So, but basically in ATCC genome portal, we are sequencing all the ATCC material, and we are putting those genome into that genome portal. So these are very high quality, like a reference reference type genome. And so far we have around like 5,000 genome available. So you can download the genome, and you can get those those genome in your research pipeline. Now, the difference between our genome portal and NCBI is that, like, okay, in NCBI genome, you may find some interesting gene within a genome, but it'll be sometimes it's very hard to get the actual physical microbes. Okay. You may not find the depositor, or the depositor may say, oh, we don't have that material anymore. There are many issues, but in ATCC is that you have the genome, and at the same time, whenever you want, you can buy those physical material, you can buy those bacteria, fungi, whatever. So that makes our genome portal very special, because we have the genome and the physical material. And on the other hand, like, say, if you have something interesting from ATCC and you don't have the genome, in that case, you don't have to sequence that. You can go to our website, you can submit a request form. So basically, it will prioritize that particular strain or whatever DCC material, and we'll sequence it. And maybe within a few months, you'll see that genome available in our genome portal. So this is a new feature we are adding into ATCC. So now let me give you some idea about our collection, what we have. So as I told you, we have a huge collection. We have around 70,000 different bacteria, fungi, virus, and protozoas. So we have around bacteria from around 200 different genera, fungus from around 1,800 different genera, and around virus from around 200 different genera. Now, giving a little bit more detail, like we have around 33,000 different mycology strains, 29,000 bacterial strains, 4,000 cell lines, 2,500 protists, and around 2,900 virus. And like say when you go to fungus, I can say that we have around like 7,900 different species of fungus, covering around 1,800 genera. And these are highly diverse in terms of species, in terms of geographical location, because we have microbes from around the world. And phylogenetically also, we have diverse collection. So that's why when I joined ATCC, and then before me also, like our job, my job in my department was to bring new things. But then we thought that what about the existing thing that we have? So that's why I started looking into see that whether we can add some application to these strains. So basically what I did, we went through the depositor information, what the depositor has said about their strains. We went to the literature, basically doing the literature search, and we started adding potential application to these microbes. So now, if we talk about like, we have different categories, but today, like I'll be mainly focusing on the food and agricultural sector. So we have identified around 1,200 different ATCC materials that can be used for food and agriculture industry, and mostly yeast, because these are the highest chunks come from all the yeast that can be used for wine and beer production. But we have other filamentous fungi, bacteria, and all those. And in terms of application, like different like biocontrol, biofertilizer, these are mostly agriculture, but other application will be more relevant for today's talk, like all this food and beverage fermentation, non-conventional yeast, like a potential cell line for cultivated meat production, microprotein, and all this. So I'm going to talk about these each group a little bit in detail. But before I go there, let's show me how you can get ATCC material. So basically, if you visit our web page, then you go to the application part, and you will see a food production and research web page. And there you have different sections, like say alternative protein, fermentation product, like oil and fat production, agriculture. And so if you click this, it will take you in different section, and at the same time, you will get this list of microbes. So basically from there, you can select whatever you need. Another best way is to use this search bar. You can search any word or any particular species if you want. You can search there, and you can get things from our website. You can buy it basically. So let's start with different categories. So first, let's talk about the strain that can ATCC strain that can be potentially used for single cell protein. As I mentioned, single cell protein, it can be the base protein for all this alternative protein production or alternative meat production. So I identified around 48 strain from 30 different genera that can be potentially used for single cell protein. Now, just to make sure, like I highlighted some of these strains from different species, but we have a lot more than this. Okay, these are only some highlighted strains for some species based on depositor information, based on literature search. So basically, you can use this list like as a tool, like you can as a starting screening material list. You can use it. Some important like bacillus or chlorella species. These are very commonly known microbes which can produce which can be used for this single cell protein production. So next, let's talk about like the microbes that can potentially produce oil and fat, which can be used for fat production. So like again, like chlorella or these are like the microalgae. These are already well known to produce oil and fat, or ERVR lipolytica, which is a oil producing yeast. We have like almost like a lot of strain for this. These things can be, you can take these and screen it and use it for your research platform, like to screen for the oil production. Next, say potential strain for fermented food and beverage production. So a huge chunks of this 250 strain from 24 genera is like Saccharomyces cerevisiae. These are all the yeast used for wine and beer production, but we have a lot of other strain, like other species and genera I have selected. These are different type of fermented food production, like traditional So these things came from different part of the world, like food and beverage production. So one advantage of these that most of these strains came from food, so food source. So it gives, when it comes to regulatory approval, it gives upper hand in that matter, because they are, you can consider that as safe for human consumption, because they were isolated from all this traditional fermented food. And some strain, like say I told about cheese production, we have around 7 species and 57 strain that are used for cheese production, like the cozy production. So all these species can be easily adapted into this like fermentation based food or fermentation industry or in the food industry. Then next come this non-conventional yeast. That's another area which is getting very popular these days, because conventionally we are using Saccharomyces cerevisiae, E. coli, or other bacillus for precision fermentation. But we have a lot other than this traditional one. So I identified around 140 strain for 24 different genera. Many of these are like Pichia or Eria, getting more and more importance these days. Like people are focusing more on this non-conventional yeast. The advantage of using this non-conventional yeast is that it can unlock a new possibilities in precision fermentation. Like say, for example, we compare two of these Eria lipolytica strain with the traditional Saccharomyces cerevisiae strain in our lab here. And we run this biolog microarray place, and we saw when it comes to say carbon utilization, nitrogen utilization, or osmotic response, they behave very diversely. Okay, so in depending on your need, what is your starting material, what is your end product, instead of using Saccharomyces, you can use this kind of Eria or other species. And when it comes to precision fermentation, the genome architecture is also very important. So how simple is the genome, how easily those microbes can be transformed, plays a critical role in like which strain you will select for your precision fermentation, what is your end product, and all those. So having more option is always better. So so basically it gives us a tool to try new things and optimize this process. So next comes the potential strain for microprotein production, which is very like, I guess, the most relevant for this presentation today, and because microprotein are getting more and more importance these days. And I identified around 82 strain from 31 different genera. These are mostly filamentous fungus. I include one Saccharomyces cerevisiae because the depositor claim that particular strain to be used as for microprotein production. And we also have this the Curon strain, and the Fusarium veneterium. You can get it from ATCC. You can like use it. You can commercialize it even. And one important aspect is that for all these strains, we have a lot of metadata available. We have the source of isolation, country of origin, year of isolation, different depositors description, various publication. And most of this strain came from various traditional food like cozy, kimchi, soya. So this can be easily adapted into into this platform of alternative protein production. And talking about microprotein, another big area which we can exploit is the potential edible mushrooms. Okay, so we in ATCC itself, we have around 13,000 different strains from 88 different species of edible mushrooms, potential edible mushroom. And I guess as people have already, I have seen many reports of people using this oyster mushroom or this chicken of wood mushroom. So we have multiple strains, so definitely people can take things from us and try that. Then another area is the food coloring. Okay, so once you produce your protein or whatever food, you don't want to add chemical colors into that. So microbes are very good source of these colors. And we have around, like say, for example, I have showcased around 180 fungal strain that can produce various pigments. And we have much more than that. Like this is just an example I'm showing you. So, so far what I have shown you, these are all like publicly available. So based on like all this information are either publicly available on ATCC website or all these publications are there. So to be honest, I haven't done any, this is not based on any research on all this. So basically what I have done, I did some data cleanup and literature search and all those. It shows that we have a huge potential. So, but now the question is like, what is limiting this widespread adaptation of these microbes? Okay. So as you know that we have more than 1 trillion microbes in the earth, and even like if you consider the known identified bacteria and fungi, around 30,000 bacteria and around 150,000 fungus has been identified so far. And it's not only ATCC, like there are a lot of other repositories. You can get these microbes from anywhere else, okay? So, but what is the bottleneck? Why people are not using this kind of microbes, or why it's not getting popular? So there are some bottleneck. One major bottleneck is the regulatory hurdle, the cost and time involved in optimizing this the process, and then getting like the regulatory approval. Then the market perception and the competition in the market also, and there are some technological bottleneck. And then another bottleneck is the availability of licensing microbial strains. So there may be many sources, like there are many repositories or many collection, but if you go there, talk, and if you try to say that, okay, can we use it for commercial purpose, then they may say that no, you cannot use it. So that's what advantage of ATCC's collection, because most of these strain or most of the things that I've shown, you can use it for your commercial purpose. And in that same context, like when I looked into the list of grass species, the grass certified species, you can see this very small number of like very handful, like say 3 species of Fusarium, and like these are few species of microalgae and Neurospora, and these are the mushrooms, the oyster mushroom, and 2 species of 2 strains of cicadi mushrooms. These are approved. So this is very handful. And if you see, when I looked into our ATCC's collection, we have several other strains from this particular species. So this can be a starting point, but I'll encourage people, like we should go beyond these things. Okay, so basically because as I showed you, we have a huge diversity, like we have like a huge collection. So how we can select the next strain? So here I have described, like we have come up with a simple diagram or a decision tree, how we can determine the safety of this microbial strain, how to use this in this context of like alternative protein. So basically you can start with identifying the species, which is obvious. First we need to identify. Then we can do the genome sequencing, because nowadays it's very easy and everyone is doing. So once we have the genome, we can do some in silico analysis. We can see that whether the genome contains some virulence factor, toxins, or pathogens. And if they don't, then we can go to the next level. We can see whether they have antibiotic resistant gene, whether they produce antibiotic, and whether these things are genetically modified. If yes, we have to do some safety evaluation. If no, it's better. And then next step, we can see whether these things with microbes came from like a food source, which is safe for animal, okay, which is always better, like a good starting point to use like the food related microbes. Then like basically we need to see that whether enough literature is available, or like say, for example, those grass certification, like say it become easy if you have a grass for a particular species, it become very easy to get another strain from the same species get approved. Or some other publications, if we have whether that publication support the safety of these microbes, and then whether these microbes can go into some other non-target animal or plant. For example, you don't want to use a plant pathogen for microprotein production or something like that. Because even if it is safe for human consumption, it may roll over to like the plant and cause some devastating disease or something like that. So once we have that, so finally we can consider these strains as a safe for human and animal consumption. So this is just a nutshell. This is very stringent process that we are trying to work on. So our aim is that we'll put through a lot of ATCC strains and microbes through this pipeline. We then will determine whether these things are safe or not. But to be honest, like ATCC, this is not ATCC's main business. Okay, we are a repository. Our main purpose is to get things, distribute things. So we are always open to collaboration. So if you are a company or if you are a researchers wants to screen things for a future strain, you are always welcome to collaborate with us. Definitely, you can buy those things from us, but at the same time, you can also collaborate. That gives us some relationship with you. We can help each other, and of course, if you collaborate, you may get things in much cheaper rate. Like we can give some discount and all those kind of things. We can have some relationship with you. We are always welcome to collaboration. So next, I'll shift our gear a little bit and I'll talk about the licensing part, because this is very important, because normal perception is that ATCC microbes are for research only, okay? And to some extent, we are creating this confusion because basically when you get a material from ATCC, you need to sign a material transfer agreement, okay? This ATCC, in short, we call it MTA. This is a corporate document, and this restrict the customer for use of ATCC product for research use only. Okay, so under this MTA, you can use the ATCC product for only research, but that doesn't stop you for commercialization or licensing. So basically, if you want to use it for commercial use, you come to us again and just request a license for that. So under MTA, what is permitted and from when when you require a license. So basically, if you want to do internal or clinical or any R&D use, you can just get the ATCC item. You can do it. You can do whatever you want to transform it, modify it. You can do it. You can do your early stage product development. You can do collaboration with your colleagues and all. You can use this ATCC material to collaborate with people. You can develop your own assay, or you can do all your QA, QC validation, your own equipment and process. So for these things, you don't need license. This all fall under the MTA. But on the other hand, if you want to produce something or provide something for fee using ATCC material, or you are standardizing a assay protocol for commercial use, or you are doing some regulatory testing, or you are releasing some product into the market, or you are giving these things to a third party as a profit, or you are commercializing and it's the product which is derived from this ATCC material and you're selling it, or you are creating a QA or QC process where you are including the ATCC product. So in that case, you need a licensing from ATCC. And why we require license? Because see, ATCC's mission is to deliver this product for research purpose. And when it comes to commercialization of this item, it goes outside our mission. And that's where we want a little bit buy-in from our customers, because to run this company, we need revenue. We don't get government support. So licensing gives us some revenue to run the whole process. And because like when someone deposit with us, it's all free. Like we don't charge for any deposit or any kind of deposit. So basically, our company runs on the revenue that we get from selling these items, or some part of the revenue come from licensing. And this licensing also protect ATCC from various aspects, like say from safety aspect or regulatory aspect, because we wanted to know how ATCC products are being used in the commercial field. Okay, we don't want like you to take ATCC product and like giving it people so that they can consume, and if something happens, then ATCC will be somehow liable. So we try to control the process, and at the same time, it also gives some advantages, also protect the depositors' right, because around 10 to 20% of this licensing revenue goes to the original depositor. So that gives some incentive to the depositor also to deposit or disseminate his or her research through ATCC. So that's why we request that you come for licensing. So so next, let's see, when does a customer need a commercial licensing? Okay, so if you are developing a therapeutic vaccine or food product like a cultivated meat, so basically you need to request a license prior to the regulatory filing. And then if you are developing a diagnostic kit or a research kit, again, you need to file a licensing prior to the regulatory filing. And if you are a CMO or CDMO, you need to request the licensing prior to commercialization of that service. Or if you are a customer developing agricultural, industrial, cosmetic, or food or beverage products. So this is the most relevant here. So basically, if you are developing this kind of alternate protein or whatever out of ATCC material, you need to request a license prior to the commercial launch of the use. So this is where, but you can come at any stage. You can come at any stage to ATCC, request a license, or to the negotiation. What will be the condition of those license? Now, the next question is how to obtain this license. So you can get this material from ATCC for research use. You can do your research, whatever you do. And then if you realize that, okay, this particular material has some commercial potential, then basically you just visit our website again, the licensing page. Then you create a licensing portal and submit a request. And once you submit a request, a licensing associate will reach out to you, and we'll negotiate all the licensing. We'll negotiate all the different aspects of the licensing. And the one question I normally get, like, how much is cost or what will be the condition? And it depends on the product to product, how you are using the ATCC product in your portfolio. So basically, like, say, if it is our ATCC product is a small part of your whole process, then definitely the cost of licensing would be very less. Or like, it depends. Or say if you have put a lot of effort into that product, you have genetically modified or did a lot of modification, then the ATCC goes down and down. So that way, so basically it depends. And overall, I will say that this is very nominal fee ATCC charge for licensing, because we encourage people to license more and more, not only for revenue, but the various other aspects. And a lot of companies are licensing with us, even in fact in this particular field. And another thing I'll say, that basically we keep it very secret, like who has licensed what things with us. We don't disclose it to anyone else unless that particular company wants it, or that particular customer wants it. So either you can disclose that, okay, we are using ATCC product for different reason, like for branding or regulatory finding, or you can hide it. It's up to you. So that's the part of the regulation. So if you have any additional question for licensing, you can directly email licensing@ATCC, and they will be happy to answer your question, or you can contact me and I can get them in touch with them. And then finally, I will always request, because this is my department, deposit with ATCC, because there are many advantages of depositing with ATCC. First is the preservation and long-term storage. And second is it support research, collaboration, dissemination of science, because the whole field, and in this field of alternative protein, we don't want to invent the wheel again and again. So if you have invented something, if you found something interesting, deposit with ATCC. We'll distribute it globally, because we have global accessibility. We have a distributor around the world. We will standardize it, quality assurance. It gives, once you deposit something to ATCC, it gives some quality assurance to that product. And it helps with a lot of like compliance or regulation, because if you are filing for regulatory approval, if you give the ATCC ID, then okay, this particular strain has this ATCC ID, it makes things much easier. And we can also some provide like IP protection, like say you can use our biorepository service or patent deposit for your IP protection. And another thing is licensing royalty. And now if you deposit something with us, and if ATCC is able to license that around 10 to 20%, like you'll get around 10 to 20% of that licensing fee as a royalty. So it gives some leverage on that one also. So to deposit things with ATCC is very easy. So basically you visit our website, select what type of service you need, like general collection. Most of these items fall under general collection. If you have something from US National Park, you can select this park service, or if you have patent related item, if you want to patent your item, you can select the patent service, or type strain. If you have identified some new species, you can select the type strain, and you just fill out a short form. And once you fill out that, then a project coordinator will reach out to you, and basically will take care of the deposit process. So I will always request you, if you have something interesting, please deposit with ATCC. That's what makes ATCC unique and special, because we rely on all this people's contribution. So with this, I would like to thank you, and if you have any question, please visit our website, or you can email. If you need anything, you can email to ourselves, or you can email me. I have my email in the on the title slide. So with that, I think I'm open to questions and discussion.
Thank you very much, Shaheen. Appreciate you going through everything and showing us the resources at ATCC, and especially the resources specifically on microbial food production. We have a few questions from the Q&A, and then a few other questions that we've found throughout. Can we go, I guess, to the one that is for your most recent slide, which is on strain depositing?
Yeah.
One of our participants asked, do you receive strains for deposit which have been passaged or evolved in laboratory, and that have genetic edits, or do you only take wild type isolates?
No, we take both. We take both. So if you have did some modification, it's always have some value. So we take both. Yeah.
Great. And then another question, could you please expand a little on your in silico safety screen mentioned in the decision tree? Are there other genes beyond antibiotic resistance that you take into consideration? And I'm guessing these would be genes like mycotoxin genes or endo or exotoxins in bacteria.
Yeah. So different. So this process, like basically this is like the first line of screening. Okay, you may miss something, but if you see that, okay, this has a lot of toxin producing gene or antibiotic producing gene, or this is a known pathogen, there's various virulent factor, it's basically not to go with that particular strain.
Great. It sounds like, again, like you mentioned, there's an opportunity to collaborate. So if anyone has any tools that can help you along with that, or suggestions for genes, that would be a good idea.
Yeah, definitely. We are open to collaboration, like, yeah.
Excellent. And then I guess a related question for me has, does ATCC look through the EFSA qualified presumption of safety list, like the GRAS list? That may be another good resource for microbes that are approved for use in food.
Yeah, definitely. Like I just took the GRAS list as a reference, but anything like if yeah, like definitely we'll go through that. We'll look into that list also. Like, yeah.
Excellent. An interesting question from Brandon Wolder. Do you accept consortia for deposit, or just pure strains in the
No, we
ATCC
use
collection?
both. We accept both. Like it's like if you have a collection, it's always good. Like especially like we can give some special collection status like those. And based on application and the current, like what is the importance of that, yeah, we'll consider all sorts of, we are open to all kind of deposits.
Excellent. And I guess another question, when depositing, can you also deposit along with genome sequences already done and annotations, or are there opportunities for people to suggest annotations for something they may be researching and then have a suggestion for something to add to the food list? Or is that going to be something that's done internal to ATCC?
No, basically when it comes to sequence, we do our internal sequence, because you see, like I was telling, like, all our product are like ISO certified, even our sequences are ISO certified. So yeah, if you have sequence it, it's well and good. We consider those to analyze, or sometimes, but basically, our motto is like we'll sequence everything ourselves so that we manage the same standard of that. And then once we sequence, we always compare the depositor sequence and our sequence. That is like another level of verification. Yeah, but we sequence ourselves. Yeah.
Great clarification. Thank you. Samaya asked, do you have any plans for synthetic gene production that customers can purchase from ATCC, like a gene synthesis, or is this going to
Yeah,
be
we are
a
doing that. We
switch?
have a lot of
Okay.
synthetics. We don't have in this food thing, like in the food aspects, but a lot of, especially all these pathogens, like especially like say like high BSL-3 level pathogens, like the monkeypox or flu.
Mhm.
We have a lot of synthetics, and synthetic is a big. All these synthetics are like a big portfolio for us. We produce synthetics which can be used for standard, for QC testing, or whatever different aspects. Yeah, we do that.
Great. Yeah, we'd encourage people to go to the ATCC website, atcc.org, and check it out. Great. I'd like to thank you, Shaheen, for the presentation today, and thank all of you for your attendance in our last 2024 Science of Alternative Proteins. We'd like to conclude now, and hope to see you back in 2025 for our next Science of Alternative Proteins. Thank you, everyone, and have a great end to your year.
Thank you. Thanks, and have a great holidays. Bye-bye.